All right, so this next lecture we are going to talk about what is referred to as the fed state. And so again, just keep in mind we're talking about this with respect to biochemistry and your body. And so in chapters 2 and 3, we are going to compare the difference between fed state and fasting state. And in general, the fed state, you're going to have a massive increase of insulin in response to eating. And this lasts approximately 2 hours. And then the fasting state is post-eating, in which you have a decrease of insulin and an increase of another hormone called glucagon. All right, so our fed or absorptive state is going to be, again, during eating. So there's going to be digestion of your eating products, whether you're eating fats, carbs, or proteins, absorption of them. We're going to have changes in our hormone levels, such as insulin and glucagon, and then we're going to have to do something with all that energy that's coming in. So in essence, our carbohydrates, lipids, and proteins are going to be ingested, digested, and absorbed. Now the major fates of fuels in this fed state, so the major fate are going to be oxidized for energy, or stored, or used for biosynthesis. Now we are going to talk about the oxidation for energy from the perspective of fed and fasting, but keep in mind it happens in both of them because your cells need constant ATP. So whether you are in the fed state, you're eating, and you are breaking down what you are eating to make ATP, it's the same process that's going to occur when you start break down energy stores during the fasting state to make ATP. So it's all the same. It just depends on what enzymes are turned on, and then what is eventually going to be the fate of those carbon skeleton backbones of your fats, your proteins, and your carbohydrates. So in essence, you have glucose, which you can take up, and glucose can form energy, so you can oxidize it. It can also go to storage, so that's where you can build up those glycogen stores in all your cells, except for red blood cells. And the glycogen stores will be stored up in liver and muscle to save for another time. You can also make fat. So TAGs or TG is fat. Glucose can also be the backbone of many different compounds. Those carbon skeleton of glucose can be used to build things. So you can have synthesis of compounds. Now, amino acids can be used directly for protein synthesis or synthesis of nitrogen-containing compounds, and it can also be used for energy during the fed state. Fats are going to go directly towards storage to make those triacylglycerols. They can also be used in oxidation for energy source if needed, especially if you're on a low-carb diet. And you can have synthesis of membrane lipids. Remember, if you go back to cell biology, all of our membranes in our cells are made up of fat. And so having fat in your diet is important to replenish damaged fat that is actually in part of your cell membranes. So let's first talk about digestion of carbs. So we have, of course, starch, which is a major dietary component. And starch is basically a polymer of glucose. And remember, it has those alpha 1,4 and alpha 1,6 bonds that we can break down. And so it's going to be a major dietary carbohydrate. Starch is going to be digested by salivary and pancreatic alpha amylases. And this large polymer is broken down into di and tri oligosaccharides. And these are going to go to the brush border of your intestine, and they are going to be broken down into glucose. And then they get absorbed. Now sucrose, as we talked about already, which is basically table sugar, and this is going to be digested by sucrase, and this is going to be broken down into glucose and fructose. And then these are going to be absorbed. And you have lactose, which is a milk sugar. And this is going to be broken down by lactase. And this is going to give you glucose and galactose. And these can be absorbed. And so you have these monosaccharides. So monosaccharides are going to be absorbed by your epithelial cells. And then these are going to go to the hepatic portal vein. And then they're going to head through the liver. And then, of course, they can go to the body. Now, proteins are going to have to be cleaved into amino acids. So you have proteases. And you have, for example, first in your stomach, you have pepsin. In the small intestine, you're going to have trypsin, chymotrypsin, elastase, and carboxypeptases. You'll also have amino peptases too. And then in your intestinal epithelial cells, you will have the ability to break down peptides, small peptides, by aminopeptidases, di and tripeptidases, and then all of these amino acids go to the hepatic portal vein to get absorbed. Now, fats are going to be a little bit more complicated because they are not water soluble. And so basically you have it's really interesting what fats do. First, you have your emulsification of your fats from your diet. And then you're going to have your pancreatic lipase. And these are going to, in the lumen, convert your tags into 2 fatty acids and monoacylglycerol. Okay, so that's a glycerol esterified at carbon 2. So you're going to have 2 free fatty acids, and then you're going to have your glycerol with a fatty acid still at carbon 2. And then you're going to take your 2 monoacylglycerols. Well, let's just put all of this. All of this is going to be bound into what we call a micelle. And a micelle is basically a fat particle, fat microdroplet with biosalts. And then basically you're going to deliver these free fatty acids and your glycerol monoglycerol fatty acid to your epithelial cells. And then your epithelial cells are going to generate tags. And then these tags are going to be packaged into chylomicrons. And then they are going to go to the liver. So you basically start with triglycerols, you emulsify them, break them down into free fatty acids and monoacylglycerols. You absorb those and then build them back into tags, and then you package those back into chylomicrons and send them off to be delivered into the body. So this is an image from your book about the fed state. And again, you have your carbs, you have your fats, and you have your proteins that all get absorbed as monosaccharides into chylomicron particles or as amino acids. And then they go to different areas of your body. So glucose, for example, will go into the liver and become glycogen. It can become energy. You can generate ATP. Glucose will also go to the brain and provide energy, and will also go to the red blood cell to provide energy. Your glucose can also, if you have excess, form fat in your adipose tissue. And glucose can also be picked up by your muscle for energy or glycogen. You have your fat, make it orange. Fat usually is going to become a tag, and it's going to go to the adipose tissue during the fed state. Your protein here becomes amino acids, and these can go to tissues, and the tissues can use them for ATP, to make proteins, etc. Or it can go to the liver, where it can feed into many different areas. Now, another thing to point out here is that when glucose is taken up by the liver, it can go to storage. So this would be store. It can be energy, or it can be stored as fat. And this particle is a very low density lipoprotein particle, VLDL. And so basically your liver takes glucose, makes fat, not that one, sorry, makes fat, packages into VLDL particles, and those go to the adipose tissue, so you store your fat. So if you take in too much carbohydrate, you're going to generate fat. Just keep that in mind. So if you have a low-fat diet and you still are taking in too much carbs, you're still going to make plenty of fat. Now, digestion and absorption. So digestion and absorption, just to recap, carbohydrates and proteins are going to go to monomers. Our fats are more complicated. They're first going to get emulsified by bile. They are then going to get digested to fatty acids, plus a monoacyl glycerol. They are then going to form a micelle. They're then going to enter your epithelial cell. They're then going to be turned into tags again, and they're going to be packaged in a chylomicron. Now, changes on the hormone level after a meal is that if you have a high carb meal, you're going to get an increase in insulin, and then you're going to get a decrease of glucagon. And depending on whether you have more of a protein, you'll have less insulin, but proteins still trigger insulin. It's not just carbohydrates that do that. And glucagon, even though we say it decreases, the levels of the glucagon levels actually stay relatively consistent. It's just that its proportion in the blood circulating decreases because you have such an increase of insulin. And it's important to keep in mind that glucagon is always going to be present because that's critical for the shift between the fed state and the fasting state. Now, hormones after a meal. So let's see, we have high carb meal. You're going to have the pancreas beta cells release insulin. And the pancreas beta cells are going to be sensitive to insulin directly in contact with the cell. Oh, I said that wrong, sorry. Glucose, they make insulin. So what that means is that the pancreas is only sensitive to what is in the pancreas. So as glucose levels decline in the pancreas and insulin levels go down, glucose levels in the blood might still be a little bit higher, or vice versa. But pancreas beta cells are only sensitive to whatever glucose is around them. And then insulin signals that glucose is available. And so this is going to tell your body that use glucose and store excess. This is kind of what insulin says to your cells. Now, after the fed state, so in the fasting state, you're going to have low insulin. You're going to have an increase of glucagon. And so the message of glucagon is glucose is not present, must be generated. So your cells quickly shift from absorbing glucose and using it to now we have to generate our own glucose. And it must be generated from endogenous sources. So this means within the cell. So this is where you can tap into glycogen, etc. Glucagon is secreted by alpha cells of the pancreas. And of course, insulin is going to be secreted by the beta cells of the pancreas. Okay, so here is the fate of glucose after the meal again. And this just goes through, again, what we've already talked about. Oh, I want to do this. I'm sorry. I'm jumping ahead. This is in the liver. So let's talk just really quickly about liver. So we're sort of taking a snapshot of this one little organ. So glucose is going to enter the hepatocytes. Hepatocytes, of course, are liver cells. And this is going to be stored as glycogen. Or it's going to form a fat. Or it's going to be used as energy. Or from some biosynthesis, depending on what that hepatocytes need. Now, the liver has a glycogen storage capacity of about 200 to 300 grams after a meal. Fat stores are unlimited. So once your hepatocyte fills up its glycogen stores, it quickly shifts and starts pushing all of the excess glucose to making fat. So excess liver glucose goes to making fat and proteins. And these are going to be packaged again into these very low density lipoprotein particles, and these will go into the blood, and then they're going to go to the adipose tissue for storage. Now, fate of glucose in other tissues is that other tissues are going to use glucose for energy. So for example, in muscle, a muscle is sensitive to insulin, and it's going to use glucose for energy. And it's going to use glucose for glycogen stores. The brain needs about 150 grams of glucose required a day, and glucose is the main fuel. It's going to be a precursor for neurotransmitters. And if your blood glucose drops, you get dizzy. Now, for red blood cells, glucose is the only fuel. And so they can only do anaerobic glycolysis. They have no mitochondria. And so in general, you have here your glucose can be taken up and generates ATP. Your muscle cells can also take in different things from the blood, such as your fats, and use that as well. Now, the fate of the lipoproteins and amino acid lipoproteins, both the chylomicrons, which come from your gut, as well as the very low density lipoproteins that come from your liver, transport fats and cholesterol to your adipose tissue. And in the fed state, your amino acids are going to be used in the liver to make serum proteins. So your amino acids go to the liver. Sorry, I think I had green. And they're going to be building proteins and also nitrogen-containing compounds. Okay, so let's talk a little bit about lipoproteins. Again, this is just an introduction to lipoproteins. We'll talk more about them as we go through the course. But we've introduced a few already. So we have micelles, which are only in the lumen of the gut. If it's in the circulation, this means integrity of gut is compromised. You should never see micelles circulating in the blood. Micelles are there to bind bile salts and your fats that you take in, emulsify them, and break them down into those fatty acids and monoacylglycerols. And then we have our chylomicrons. These are formed by your gut epithelial cells. And they are a package of your dietary tags, cholesterol, and they have some proteins associated with them. And so chylomicrons are made by your gut epithelial cells, released and picked up by your liver. VLDL are lipid particles made by liver to store excess fat and cholesterol. Now, each of these particles we're going to talk more and more about, and they are going to have association with cholesterol levels, of course, and plaques, atherosclerosis and plaques. But here's just a little quick introduction of the ones we've been talking about here. Okay, so next up is our fate of our amino acids. So our amino acids, of course, are going to have our nitrogen-containing compounds, and the liver is going to use amino acids for the synthesis of serine proteins. So liver will use to make serum proteins. Now, the liver makes a lot of proteins that are in our serum. So the number one protein is serum albumin. And serum albumin is the most abundant protein in your serum. And serum albumin is basically there to help things get through your blood. So it's going to bind to hormones, it's going to bind to vitamins, it's going to bind to all kinds of things to make sure that they don't get degraded or broken down in the blood. Now, the liver can also use your amino acids to make its own proteins. Liver is a major metabolic organ in your body. It needs to make detoxing proteins so it can handle alcohol, drugs, whatever you throw at it, antibiotics. The liver is going to make all of the urea to dispose of excess nitrogen, so it needs all those enzymes in the urea cycle. Liver is going to be a major source for just fuels for making glucose, building up glycogen stores, etc. So it's going to use it for all of that. The liver is able to convert amino acids to glucose or ketone bodies as needed, and then it's able to take the nitrogen and convert it to urea. So many amino acids go into the blood, and tissues can use it for protein synthesis or energy. And then proteins are going to undergo turnover. They're going to break down into amino acids and be released into the bloods. And the one thing to keep in mind with protein is you have to deal with the nitrogen. Okay, and so that is in conclusion of how we ingest carbohydrates, lipids, and proteins, what happens to them once you absorb them, and some of the fates of them during the fed state.